Thank you, Dr. Opine and Dr. Rainey, for inviting me to this grand rounds. And it is an honor to be able to take this opportunity to speak to a wider audience in this department. And thank you all for your coming to this talk. And today's talk will be addressing the resurgence of pertussis whooping cough and local epidemiology. Bordetella pertussis is known to be an aerobic gram-negative coccobacillus that colonizes respiratory tract of humans. So far, no animal reservoir has been found, and this organism is unable to survive in the environment other than human body. And in microbiology terms, or old microbiology terms that we still use, this bacterium is known to be biochemically inert, meaning the organism does not perform much of the sugar fermentation or make color changes in terms of pH when used amino acids or other biochemical compounds as substrate to declare its identity. That's by old traditional bacterial identification method. It is known to be a very slow-growing organism, fastidious. And then even under optimized condition, laboratory condition, that is, specialized media, such as Regan low media that we use at Children's, that's composed of nutrients that included high concentration of horse blood or horse serum, and with charcoal to counteract of inhibitory actions of substrates that's present in respiratory specimens. And it also contained an antibiotic agent to select out outgrowths of oral flora. And this agent is a cephalosporin compound called cephalexin. So with that, we know this organism is intrinsically resistant to beta-lactam drug. And it is the drug of choice for treating this infection is actually macrolides, such as erythromycin or Z-Pak that everybody know by the name for azithromycin. So in the old days, I heard from my predecessor, if you need to culture this bacterium, the physician who orders such culture would come to the lab, and the lab will draw your blood to make high serum content, which support the growth of this organism. In such a high serum concentration and nutrient condition, it takes about 3 to 7 days to have a visible, viable organism to develop a visible colony. So by the time you make the diagnosis, the patient is long gone, and it's only served as a confirmatory test at the best. The transmission of Bordetella pertussis is known to via aerosol droplets that directly coming from the respiratory tract of infected individual to susceptible host. And this is known to be, this organism has the host restriction to humans, and then it is the etiological agent of whooping cough or pertussis. This organism is able to adhere and proliferate on the ciliated epithelial cells, and is able to invade alveolar macrophages. And this organism is also known to produce a whole array of virulence factors, including pertussis toxin, adenylate cyclase toxin, filamentous hemagglutinin, fimbriae, tracheocytotoxin, and perdactin, also dermal necrotic toxins, and which can result in systematic infections that are severe in infants. So the clinical course of whooping cough can be separated into three stages. The first stage is called catarrhal stage. It's characterized by runny nose, irritating cough, and low-grade fever. And this stage is known to be most contagious. And usually the organism remain viable and culturable in the first week after the onset of symptoms. And post first week of infection, the organisms are hardly cultivable, and PCR has improved, molecular testing so far has improved its sensitivity and to be able to detect bacterial DNA even after 2 weeks of symptom development. The second stage is called paroxysmal stage. It's signified by rapid cough as the patient choke out air, followed by a whoop sound, which is due to the rush of air into the lungs against swollen, inflamed, and narrowed glidus. And this organism, the infection of this organism has clinical significance that includes the encephalopathy, irreversible brain damages, pneumonia, and collapsing lungs. Also hemorrhage due to the mechanics of virulent cough that causes increase in intrathoracic pressure that ruptures blood vessels and cause hemorrhage that can be even noticeable on the face of a child. The third stage of this infection is called convalescent stage, which can last 2 to 3 weeks. And then it usually has lessened cough spells, and also, but it's still susceptible to secondary bacterial infections. Pertussis is known to be a vaccine-preventable disease. And then since its first identification and its association with whooping cough in 1906 by Julius Bordet and Octave Grangeau, shortly, just a few years of time, the first formally activated whole cell vaccine was licensed in the US in 1914. And in 1942, based on the work by Pearl Kendricks, the first packaging of combination vaccine that included whole cell pertussis vaccine and 2 other bacterial agents that are diphtheria and tetanus toxoids were used for mass campaign of vaccination in the US during the late 1940s, the early 1950s. However, this vaccination was a great success in reduction of pertussis infection. However, in the 1970s, there were reports of infant death and neurological disorders reported that's associated with pertussis whole cell vaccine. And it was in 1975 that Japan was the first country that suspended whole cell vaccine and replaced it with acellular vaccine that's a combination of 2 or 3 Bordetella protein components that was developed by a Japanese scientist, Yuji Sato. The mass application of acellular vaccine in the US was in application during the late 90s and early 2000s. Currently, there are 2 packages of pertussis acellular vaccine that are in combination with diphtheria and tetanus toxoids. The 2 different packages are actually very similar, except the dosing of each components are quite different. The infants and children vaccination is composed of higher concentration of pertussis protein components that included inactivated pertussis toxin, filamentous hemagglutinin, protactin, and sometimes a different maker of the vaccine would include fimbriate component. The adult vaccine has much lower dosing of each of the components. And then the vaccine schedule is for children is 5 doses at 2, 4, 6 months, and 15 months, and at 5-year-old. Since the introduction of vaccination in the 50s, the pertussis epidemiology has completely changed. So this graph shows you the years prior to mass vaccination against pertussis. The total cases reported to CDC and in CDC records ranges from 100,000 to 250,000 per year. And subsequently, due to the mass application of vaccine, the total cases have dropped to less than a couple cases a year per 100,000 population a year. But since the early 90s, there is a steady increase of pertussis reported to CDC that has reached 10% of the pre-vaccine era. So this is from CDC website, which summarized the states with incidence of pertussis the same or higher than the national average incidence numbers ending September 20th of 2012. And our state was placed on the 3rd place, which is not a pretty place to be, at 58.1 cases per 100,000 population. So here is a specific MMWR publication focused on the pertussis epidemiology in Washington State, 2012. And this graph shows you by the whole year separation of weeks, 52 weeks. The red bars represent 2012 cases per week. And then the fading blue bars represent the same case numbers in the same weeks in the year of 2011. There's actually 13 times increase in pertussis case numbers in Washington State this year above last year. So laboratory testing played a significant role in identification of these outbreaks. And then our laboratory was acknowledged for its pertussis testing that is able to speciate different Bordetella species using multi-target PCR assay. And here is a graph showing the age distribution in Washington State during this outbreak. So the high-risk attack age range is still among the infantile, under 1-year-old population, and also we have a peak attack rate in age 10. And then this graph also shows you, reflects the application of acellular vaccine and transition from whole cell vaccine. So this dark background reflects the case numbers that's in transition from whole cell vaccine to acellular vaccine. So people aged greater than 16-year-old had a significantly lower rate of infection than obviously the acellular vaccine age group. Here is a graph that's showing the incidence per 100,000 population by age distribution. Also, this dark blue bar represents US incidence except Washington, and then the light blue bar represents US incidence. So the age distribution is very similar to our age distribution in this spring outbreak. Pertussis test data from children this spring has contributed significantly to this data. And then we have in less than 6 months period received greater than 6,000 specimens. And the positive cases peaked in May. And then positive rates testing rates over the same period of time remained similar to overall rate of 3.7, so range from 3+ to 4+%. And the age distribution, if we use the vaccination schedule of less than 5 months, who would have had 1 dose of vaccine, and age between 6 months to 2 years, who would have had 3 doses of vaccination, and 3 to 5 year would have had 5 doses of vaccination, and then 6 to 21-year-old would have had included the booster vaccine that's recommended for teenagers. So the high-risk case number are still interestingly in the range of 6 to 21. The case rate, positive rate by age, is still in this age group. And then the pertussis epidemiology this year actually overlapped a respiratory viral season. And we published this testing data on our website, and it's updated weekly. So this grass green line represent the flu A. It peaked in April, whereas this ocean blue line represents pertussis cases that peaked in May, where other viruses are in the mix. In this year, we started using a new viral detection system designed by Idaho Technology. It's called FilmArray. It simultaneously detects many viral agents that included adenovirus, coronavirus, and human metapneumovirus, rhinovirus, flu A and B, paraflu viruses, and RSV. So I asked the question, are we able to tell pertussis from respiratory viral infection clinically? So upfront, in this spring, we offer both pertussis PCR test and also respiratory viral PCR test, which is a 2-hour turnaround rapid test that replaced the traditional FA detection. And then our pertussis multi-target PCR positive rate was at 3.7, which was represented 188 cases over 5,000 plus tested specimens or patients. In those, 175 were due to Bordetella pertussis, 11 were due to Bordetella parapertussis, and 2 were Bordetella home CI. And out of all those PCR positives, we were able to obtain 48% from those positive samples organisms, viable organisms. And then we could recover about half of the parapertussis, and we actually recovered 1 home CI. And pertussis, and a subset of that 5,000 patients included who received the dual testing of pertussis and Idaho respiratory viral PCR. And of those 528 patients who had dual testing, 17 had Bordetella pertussis, and 1 had Bordetella parapertussis. And since the April of this year, Idaho PCR has gotten additional agents cleared by FDA that included Bordetella pertussis and Chlamydia mycoplasma. So with residual specimen that we saved from respiratory viral testing using the viral transport media, we were able to test 13 out of 15 Bordetella pertussis and confirm and using this system. So the sensitivity of this test compared to our multi-target PCR was 87%. And then the other 2 Bordetella species cannot be detected by design because this test only target IS481, which has a homolog in Bordetella home CI. And the viral co-infections actually are very common. So here are the 13 cases who were tested positive for pertussis. And out of the 13 cases, by majority, many of them have co-infections of rhinovirus. 1 had RSV, paraflu-4, human metapneumo, and then 4 patients actually had more than 1 viral agent. So by just looking at the data of these 2 tests, we can predict that separation of pertussis from viral respiratory infection is difficult clinically. So co-infection is very common. So why multi-target PCR is more sensitive than single-target PCR? This is a paper that we published in 2007, which summarized our test from 2002 to 2005. So we use a multi-target PCR assay, which employs 4 Bordetella pertussis targets, and we use a consensus discretion criteria to determine test results. So we have to have more than one target positive to confidently call it Bordetella pertussis. And by using multi-target, we are able to separate out parapertussis and home CI. So if we say use multi-target assay, we detected 309 cases. And then out of the 309 cases that were detected by IS only was only 79.9%. By pertussis toxin promoter targets, it was 76%. And by REC-A or porine gene was 74%. So if you combine 2 targets out of the 3, you achieve much higher sensitivity. So having multi-target PCR has the benefit of increasing the sensitivity of bacteria by two principles. So we all know that bacterial genome has target sensitivity. For instance, IS481 is 200, has each organism, each genome of Bordetella pertussis has 230 plus IS481 copies, and then pertussis toxin and RAC genes are single copy per genome. However, IS element is a non-coding sequence. And then during the early phase of infection, if you catch one organism, you catch 230 copies. So it's quite sensitive. But over the course of bacteria disintegration and degradation, I hypothesized that the genome degradation during infection is not random. And later on during the course of infection, actually the sensitivity of single copy genome target performed just as well as multi-target genome, multi-copy genome target. So, and then there's an analytical sensitivity and specificity as well, because we are counting on a PCR to work with primers, nucleotides, polymerase, buffers, ions. So if one of them is not in control, the PCR may be falsely negative. Therefore, populating a reaction into more than one gives you the opportunity for all these variables, and that improves analytical sensitivity and specificity. So this spring, the pertussis outbreak was a really test of our surge capacity and disaster preparedness. I guess we've heard many talks on the topic of disaster preparedness and mass casualty in terms of natural disasters such as Katrina or earthquake, and hospital readiness and operating room readiness for taking care of mass casualties. But in this case, pertussis epidemiology was a mass casualty that has to be taken care of by a laboratory diagnosis. And for infectious disease diagnosis, turnaround time is the key. And then for infection prevention and infection control. So during this outbreak, we have experienced a surge and disaster. And then we had to quickly come to PCR automation, and we have to come to our hospital budget committee to support us for additional thermocyclers. We have to stockpile PCR primers and reagents because we're running them like water. And staffing model has to be changed because we're experiencing on days exceeding 150 specimens per day. And we have to push them out in a timely fashion. So we, at the time, staffed our molecular section with 5 to 6 technologies with rolling scheduling starting from 4 a.m. and covering till 8 p.m. to push out specimens. And other sections of the lab had helped in specimen receiving and log in. So it was a teamwork for the surge. So the next topic that I'm going to deal with is why do pertussis vaccine fail? So this is a paper published recently by a physician who was intimately involved in vaccine trial in the 90s. And the first 2 points he listed was regarding to the overestimation of efficacy and then the flaws in terms of case definition. The other factors are are due to other species, possible other species that cause pertussis, like whooping cough. It is known in 2011 in Columbus, Ohio, they have experienced a similar pertussis outbreak, and because they used a commercial testing system that target IS only, the residual specimen was sent to CDC. And of those residual specimens, 1/3 of the specimens were actually Bordetella holmesii. So CDC was very pleased with our assay to be able to speciate the 3 different species. And then the 4th point was lack of initial vaccine lacks the initial potency, and also overall decay in antibody over time, and an incomplete antigen packaging, and incorrect balance of antigens in the vaccine combination, and also genetic drifts in Bordetella pertussis. So basically, the well-accepted reasons behind pertussis, the failure of pertussis vaccine was due to, first, waning immunity. Secondly, vaccine efficacy was not complete, and also public concerns for immunization, also pathogen adaptation to herd immunity by antigenic drift. So I'm going to address the following 4 points one by one. So the waning immunity states for the no lifelong immunity of pertussis vaccination. The immunity wanes within 5 to 10 years after completion of childhood series. There are a growing number of infections among adolescents, adults that are underestimated, and 20 to 30% of those age group having prolonged cough may indeed be due to pertussis. And then, so therefore, the recommendation of booster vaccines. And pertussis vaccine efficacy, the acellular vaccines clearly shows reduced efficacy and short duration of protection, and provides initially only even if you completed the whole series, high childhood series, the protection was only 80 to 90%. And it gradually decreases to 50% after 4 years, and completely gone after 12 years. So there is also public fear of immunization due to a retracted famous paper by Wakefield that associated vaccination to increase in to autism and bowel disease. So the negative publicity resulted decrease in immunization rate, and therefore a growing number of infections. And then lastly, pertussis vaccine are protein components, and these are the 3 main components used in all vaccine packages. And then by sequencing of these protein encoding genes, we have found that the genetic drifts has happened in all these protein encoding genes by point mutations. And therefore, it is hypothesized that the emergence of pertussis perhaps is in part due to antigenic variation, an evasion of immunity. So the conventional wisdom calls for a better vaccine. And then I'd like to think out of the box and see what else have not been taken into the consideration. So there's a paradox. Evolution of niche-specific or host-restricted lifestyle are found in both beneficial symbionts in animals, hosts, and in deadly pathogens such as pertussis and group A strep and group B strep, Staph aureus, name it. So why do host-restricted symbionts lose virulence and provide benefit to insect and many marine animal hosts, while human host restricted pathogens retain virulence and transmissibility. Microbiologists always focus on microbes. We forgot the other part of the equation, that is the host. So the ever-changing host environment and an associated microecology has to be taken into consideration. So contemporary medical practice uses vaccine, and that's a huge success in advanced host immunological defense. And secondly, using antibiotics to sterilize infections, especially acute fulminant infections. But selection for drug-resistant microbes and cause dysbiosis of the host can also be a consequence. Secondly, agriculture practice of antibiotic into the food feed and dietary practices have changed. Climate change also can change the survival and redistribution of disease vectors. And fourthly, increase in population size and density can also contribute to the infectious disease equation. And natural disasters or political and social unrest, such as cholera outbreak after earthquake in Haiti. So host adapted microbes are protective. We understand now the host adapted microbes are protective of our health and integrity. So our species is not a single species. We are a composite of many species. Bacteria symbionts on our surface of our skin and interior of the gut and the respiratory tract are now understood to be protective of our health and integrity. And in the past, the medical books are written in such a way that our skin, our mucosal membrane is our first line defense. However, now we recognize our human microbiome is actually our first line defense. So we all know that the broad spectrum antibiotic treatment have resulted in dysbiosis of the gut, and therefore predispose the host to C. difficile enterocolitis. And then there is now unconventional practice of fecal transplant. In the past, it would have been thought of a witch potion. So the green witch, the West now has become the good witch of the East. That's what I learned in American culture through my kids, The Wizard of Oz. And then The Wicked will be on Broadway show soon in Seattle. So I have also learned from studying cystic fibrosis and Pseudomonas in cystic fibrosis. I have a paper recently accepted by antimicrobial agents and chemotherapy where I demonstrated that the host-adapted microbes, in this case Pseudomonas, are protective of our host integrity. This is the opposite of what everybody thinks, because Pseudomonas kill CF patients. Indeed, actually, they are on their way to symbiosis. They are symbionts. We all know that when cystic fibrosis patients who are chronically infected, their organisms become very slow growing. They lose. This is a 24-hour growth on Mueller-Hinton agar. This is the susceptibility agar that microbiologists are very aware of. A wild-type colony after 48 hours is this big, and a highly adapted CF Pseudomonas are very small and slow-growing. And then from a single patient, no matter how divergent their phenotype is, or how resistant or susceptible they are to antibiotics, they are clonal. They are generated from a founder clone and diverged into different phenotypes. And as soon as the patient has organisms that displaying divergent phenotype and susceptibility, they stop acquiring wild type Pseudomonas. So in all chronically infected CF patients, you never see a wild type. You will see they continue to carry these organisms. Although they try to be your symbiont, maybe they want to become your mitochondria. However, the evolution is given such a short period of time because the evolution will abort as the patient demise, and the damage has been done, and this cannot be reverted. So actually, my observation suggests that these adapted human microbes are protective of you from getting wild type, more virulent strains. And I can show you one more compelling figure, which I use the minimal media, which contains glucose and salt only. And we know wild type Pseudomonas is able to assimilate sugars into 20 amino acids and grow very well on the minimal media. But CF adapted Pseudomonas, many of them lost their ability to synthesize all 20 amino acids from one of the patient isolates that I streaked in like 2 eyebrows back to back. One wild type underneath and the CF adapted, host adapted organism on the top, and let them grow in 2 weeks. And when the wild type migrate towards growing and migrate towards the host adapted CF organism, although this organism is unable to grow, it seemed to have a contact inhibition to deter the growth of a wild type. This is on a petri dish, not even on a CF host. We can't think of anybody, even a CF host with defective chloride channel. Their lungs are not a thin-walled flask of culture media. There's enough neutrophils and enough antimicrobial agents in it. And then we leave our imprints on these organisms. And sure enough, there's another publication from actually 2011, last year, that demonstrated also oxytrophic mutant of Staph aureus interferes with nasal colonization by wild type. So this makes me think, resurgence of pertussis, there are probably more factors contributing to this new epidemiology, and may probably contribute to our new understanding of their pathogenesis and prevention. So I think antibiotic use in modern medicine may have implications in perturbation of our biodiversity, and that, therefore, dysbiosis and human microbiome may result in immunological deficiencies or immunopathology and susceptibility, and then dysbiosis may really cause the host to be susceptible to more virulent organisms. And secondly, when a pathogen invades a host, it exploits a new nutrient environment or host niche, it has to not only break down the host defense, it has to break down its host protective flora. Therefore, I propose alternative vaccine approaches. One is to fend it off. That is the traditional method to strengthen ourselves, the host, and our holistic microbiome. This is new. Where did they go when we need them? So by thinking of more of protective microbiome, we should study their interactions and their protectiveness to our health. And secondly is to invite them in. That is to identify a core molecular toolbox necessary for host association without damage. So this practice was already not new, because a live attenuated vaccine has been used in many infectious diseases. So we should probably look into a Bordetella species that is able to colonize our airway, upper airway, and is able to protect us from wild-type virulent pathogens. So with that, I'd like to thank a student, Luan Nguyen, who received her master's degree last year at Lab Medicine, who did the sequencing of 80 isolates from 2005 to 2011 that showed antigenic shifts. And I'd like to thank Dr. Rutledge and Dr. Mike Ashton for their support of our laboratory during the pertussis outbreak and give us the speed, fast track of instrument support and thinking out of the box and help us to create staffing models such that we can cover the longer day and push out more specimens, and for their leadership. And I'd like to thank Dr. Jeff Duching at King County State for his close communication with us, and Ches DeBalt at State Public Health Lab. And most of all, I'd like to thank microbiology scientists at Seattle Children's Microbiology Laboratory. And then they've done a fantastic job in getting the ready and then to meet the patient care needs. And with that, thank you for your attention, and I'd like to take questions. Yes, April.
So a quick question about whenever the outbreak, whenever it was recognized that there was an outbreak going on, was there a questionnaire that was handed out to determine when the patients had received their last booster, or was that information collected to your knowledge at, for each patient that the sample was sent?
Yeah, the question is, along with the sample submission, whether there were immunization records coming with it. I have to say no, because it was it was an outbreak situation, and specimen flooded in without any additional information. We could see probably by patient last names, and sometimes there's a family outbreak involving all siblings and parents. Other than that, we don't have immunization history. Yes, Susan?
Do you have any plans to pool, because you were completely slammed this year, and we don't know what, it's going to be like this year. Do you have any plans to pool your specimens maybe to do some sort of preliminary screening? Because it's a pretty involved assay that you guys are doing.
Correct. So, thank you. That's a very good question. I'd like to repeat the question. So, are we ready for the next outbreak and surge? So, can we pool the specimen and then for a primary screening, then further testing the number of positives? No, we have decided not to do so, because pertussis is such a dynamic disease. The agents are not, the copy numbers are very unpredictable, and specimen qualities are unpredictable. We actually have an internal control target where we would have to have the human beta-actin positive to be able to report true positive. If it is human target negative, we would have to call it indeterminate, which would suggest it's a poor specimen. And then you would have to, if clinically indicated, you would have to resubmit a specimen. And then you lose sensitivity by pooling. We know that. Thank you.
Is it known whether suffering from pertussis infection is any more protective immunologically downstream than the whole cell or the acellular vaccine?
That's a very interesting question. Let me repeat it. Is a clinical infection more protective than a vaccine prevention? And actually, I have not seen a very good systematic comparison. But it is known that the pertussis infection does not prevent you from future infections. That's as far as I know. Yes, please.
Shwin, your positive rate of 3%, I don't know how to interpret that in terms of, is that reflect overutilization or appropriate utilization? And are there any clinical screening guidelines that you provided that you felt either were or weren't followed that would have resulted in a higher rate of positivity among samples submitted?
Yeah, so the question is, the low positivity rate of 3.7% may reflect overutilization, and would we come up with a clinical recommendation such that we can test targeted population? So this is a very good question, because in respiratory season, sniffles and runny nose and cough are very common, especially in children and infants. They can't really tell you where it hurts. You know, my head hurts, or my chest hurts, or my throat hurts, and then it's almost like it's up to the clinician to recognize clinical indications and signs. And there are CDC characterization of clinical diseases and possible diseases. There are categories of clinical characterization. And I was hoping that everybody, it's a very, very well written, very informative, available to the public on the CDC website. But I think it's probably underutilized, and then over testing is a problem. Even with the respiratory viruses, we see a great overutilization. There are repeat testing of the same rhinovirus 7 times within 3 months period. And the repeat of pertussis is much less. However, the positive rate is so low, I do not know whether this is a sampling problem or it is also a public awareness because of pertussis epidemiology. And we had it, you know, on all the TV news and in the public media. Therefore, a public awareness and overreaction could be part of it. Yes.
I was surprised that they got FDA approval. A number of companies seem to be kind of moving that way in addition to FilmArray. So do you know if the CDC is planning on making any recommendations with regards to detection of pertussis by these FDA-approved platforms? Because clearly, I mean, they're not as, I was very familiar with your assay and how rigorous it is, and I was kind of surprised that they let that fly. Do you know if they're planning on making any recommendations with regards to backing stuff up that's negative for culture or whatnot?
It has not been. Yeah, the question was why CDC fast-tracked this approval of pertussis with a single target, which seemingly is insensitive and could potentially be misleading, having a lot of false negatives. So as far as I know, by talking to the company, they are not being recommended to add other targets and to be able to speciate other potential pertussis-like illness. So I guess earlier this year there was a publication on speed up of FDA approval of clinical diagnostic kits that will have benefit to patient care. I wondered if this is part of the change of policy, therefore allowing more rapid development of test kits. And then, of course, with this policy change, therefore, there will be some imperfect testing being on the market. Yeah. Thank you very much.